in the laboratory of Dr. Neil Schneider. And then she was back to Germany to do a junior group leader position under the laboratory of Professor Langenhans in Leipzig. And today we will hear about her research, which was recently published in Nature Medicine at the very beginning of this year. Before going on, we would like to know something science related also maybe to some experience you have in science, or something that it was a really striking moment, or somebody who really had a really impact that you became a scientist.
Oh, hi everybody. Thank you so much for the nice introduction, first of all. As I commented to you before, I would like to say that one of the scientists that probably had more impact on me when I met him was Ben Berres, which was also a neuroscientist working mostly in glia cells and all these astrocytes stuff. And I think his story was very interesting. He also wrote a book about it. And yeah, I found him very inspiring. And that's why when we talk about it, I mention him, because I think his legacy is very important in neuroscience.
Absolutely. Okay, with that said, I think we can start with your presentation.
Mm-hmm. I'm gonna share it and you can tell me if you can see it and hear me well.
Yes, I think it's perfect.
Okay, good. So yeah, thank you very much for inviting me to give this talk here today. Today I'm gonna talk, even though I'm in Leipzig, I'm gonna talk about the work I did at Columbia University, which was recently published. It's about ALS. We work with amyotrophic lateral sclerosis. And I think it's a good presentation for all scientists, and especially young scientists, because they can see that what we do in the lab can be translated finally to clinic. And you see like the final goal is getting closer and could be accomplished in a short period of time. So that's why I name it From Bench to Clinic, because you will see that we have a lot of mouse work, but also patient data in there. Sorry, I have to admit somebody to go forward with the presentation. So first of all, what is amyotrophic lateral sclerosis? You might know Lou Gehrig, people especially from the US, or Stephen Hawking, or even Gleason, the football player. They all share that they have have ALS. But if you still don't know what ALS is, you might remember the Ice Bucket Challenge, who took place in 2014, and a lot of celebrities took part of it. And the goal was to raise money and awareness about the ALS, about amyotrophic lateral sclerosis. And this money was all given for research. So amyotrophic lateral sclerosis is a motor neuron disease. And you have here how it develops. So we have degeneration of motor neurons in the degeneration of neurons in the motor cortex, and this one sends projection to other neurons here in the spinal cord. So we can have, as you may have seen in these patients, degeneration of muscles like the tongue, the arm, or the leg, depending on in which region these neurons are degenerating. Here you see a section of the spinal cord, and here we get this degeneration of the motor neurons. We also get in the level of the muscle degeneration of what is called neuromuscular junction. So the motor neurons send projections to the neuromuscular junction, and these ones get innervated. When we have ALS, this doesn't happen, and this innervation is being retracted. So it's characterized by motor neuron degeneration. Paralysis is normally an adult onset, even though we have kids with ALS. And the average life expectancy is between 3 and 5 years. We have 10% of cases that are familiar. That means that we know the genetic cause, that there is a history in the family. And then we have 90% of the cases that are sporadic. Either we don't know if there is a family history or they just happen in the patient. The most important thing is that there is no effective treatment so far. So there are several challenges facing ALS. One of them is the amount of genes that show mutations and these develop to ALS. You have here a timeline since 1999 we have been discovering different genes with different mutations that cause ALS. And at the beginning, we thought we could find a common pathway for these genes. And with this common pathway, we could generate animal models and translate this to therapeutics. Lately, we have seen this not the case. Every of these genes act differently. It's involved in different processes. So we need custom-made therapeutics for each of these mutations. So we focus on the gene called FAS in sarcoma, which is an RNA binding protein. And we generated animal models for it. And finally, our goal was to translate this to therapeutics. Why did we pick FAS? First of all, FAS mutations cause early onset. You can see it down here. So when we have one of the mutations for FAS, we see patients between 10, 11 years till 40 years. So it happens very early in young people. So imagine that you are completely healthy and out of the sudden you start feeling weak, and then you get degeneration, and this goes very rapidly. So in a couple of years, then they die. So that's why we decided to study these mutations. And here you see how FAS is characterized. So we see mutations all over this gene. And here you see which ones we picked. We picked this one that is an skipping of the exon 14, because it's known to be an early onset mutation. And we picked this one, which is a point mutation that it's called P525L mutation at the very end of FAS. So first of all, what we did was to generate knockout lines for this gene. And how we did that, we fluxed 3 of the exons. And when we do 3 recombination, we can skip these 3 exons and then generate a complete knockout. We checked that by Western blot, and we could see that it was indeed a real knockout. And we wanted to see if these animals would show any hallmarks of ALS, for example, motor neuron loss or denervation. So first of all, we thought we checked where we could find FAS, if it was there or it was gone. Here is an hemisection of the spinal cord, and in gray we have the motor neurons, the ones that get degenerated in ALS. And as you can see, FAS is very characteristically expressed in the nucleus here, in the wild type control. And when FAS is absent, we don't get any expression, meaning that our knockout was indeed a real knockout. When we check for motor neuron loss, when we counted these cells, we didn't see any loss. And when we check in the muscle, we also couldn't see any denervation. Then we decided to generate knocking lines, meaning or mimicking the human disease, so point mutations or exon skipping, like I showed you before. And we generated this one here that has a mutation in the P517L site. This is the equivalent of the P525L site that I mentioned before. And then we generated here this mouse model that skips the exon 14. We check, first of all, where FAS was expressed. And we could see that, as I said, in the wild type situation, FAS is normally expressed or highly expressed in the nucleus, even though it shuttles from nucleus to cytoplasm. And when we have the 2 mutants and we generated 2 specific antibodies against them, we can see that the mutant protein goes to the cytoplasm. That is similar to what happened in humans. So we also wanted to check in postmortem tissue what we could see in humans. And here you see that. So here we have our FAS normally highly expressed in the nucleus, and even though I said it shuttles from nucleus to cytoplasm, and when we have the patient, you see that all FAS is accumulated in the cytoplasm. Here is our nucleus, and it's accumulated in these characteristic aggregates. So it's very similar what we see in mice, meaning that these animals were indeed showing some similarities with the human situation. We also check if we could see motor neuron loss. As I said, here we have the hemisection of the spinal cord. Here is these are our motor neurons. And we check if we could see loss. In the wild type situation, we don't see any loss, but then in both mutants, we could see that after 1.5 years, they started to show motor neuron loss. We also check the muscle, and here we have what is a neuromuscular junction, what I showed before. And this is the presynaptic part, this is the postsynaptic part. When we have a complete, fully healthy innervated NMJ, we see this colocalization. We have a partially denervated, you see that the axon is starting to retract, and we see only the postsynaptic part. And when we have a denervated neuromuscular junction, you see that there is no axon anymore. So we only see the postsynaptic part. We check if we could see some degeneration there, and we could see that after 1.5 years, we saw NMJ denervation in both animal models as well. So it was very nice because we were recapitulating what we could see in the humans. So we decided to combine these two mutations and see how the animals survive, if it was a toxic gain of function, if we will lose the function of the protein. So as you can see here, when we have a homozygous line for this mutation, the animals don't survive more than a few hours. When we combine both of them, the animals survive around 20 days. And when we have the exon 14 homozygous line, the animals survive a little bit longer, meaning that there is a degree in toxicity between mutations as well. So you can see also that here we have, in combination with the knockout, we see the median of survival is half of what we see in the wild type, and then it decreases depending on the mutations we have. So we can see that we get a mutant gain of toxic function. We also decided to combine these mutations, but not, since we saw what I showed before, that this one here died after 20 days, we wanted the animals to live a little bit longer. So we didn't want to have both mutations expressed in every cell in the animal. So what we did is we had the P517L mutation expressed everywhere, and the exon 14 mutation only expressed in the motor neurons. Then we checked denervation and motor neuron loss. These animals survived longer, and we could see denervation as well as motor neuron loss. We decided to study these animals in the future because they show denervation and motor neuron loss earlier than the other animals, than the heterozygous animals. And this would be very easy for us when we have to do drug studies or something like that, to check earlier in development instead of waiting 1.5 years for the results. So with these results, they were very promising. We contacted Ionis Pharmaceuticals and we asked them if they were developing any drug against FAS. They say they had an antisense oligonucleotide and they developed that, and we could test this in our animals. How this works? Here we have our DNA, and this is translated to mRNA. When we have our antisense oligonucleotide, it's a piece of RNA between 18 and 30 nucleotides. It binds to our mRNA and it destroys it. So we don't have protein synthesis. The protein that it will be created after that is not longer there. So you can see it also here. We have the binding of the ASO to the RNA, and then the protein will get, or the RNA will get destroyed and we won't get protein. So we started to inject this in our animals. We did the ICV injection. So you see the injection site here, or you can also see it in this scheme. And what we saw was that when we had these injections in the animal, the amount of protein in wild types as well as in mutant, it was decreased. So meaning that the treatment was working. Then we check if the how FAS was located in these mutants. So as we saw, FAS is mislocalized to the cytoplasm, but after administering this drug, FAS was mostly located in the nucleus, meaning that this treatment was also working at the cellular level. And we also checked if we could see motor neuron loss or denervation. And after the drug, we could see that the motor neuron loss was rescued, and the NMJ denervation was rescued up to 6 months, meaning that at this point of time, we couldn't detect any drug in the system of the animal anymore. Since this worked very nicely, at that time, we had a patient that had the P525L mutation, and we tried to get FDA approval to check how this drug would work on her. So the patient was, she had, as I say, a fast P525L mutation, and at the symptom onset, she was 25-year-old. Her twin sister died from ALS at the age of 17 with this exact same mutation. And the treatment with the ASO began 6 months after disease onset. At that time, she was already paralyzed and with artificial ventilation. So here you can see how the disease progressed on her. So at the beginning, before the treatment, it was very rapid. It went really fast. And after administering the first doses of the ASO treatment, you can see that the progression decreased. There were 12 doses administered in the time of 10 months, but unfortunately, after 1 year of treatment, the patient died. Thankfully, the family donated all the tissue and we could check how this drug worked on her. And as you can see here, we check the total protein and you can see that is all decreased after administering this drug. We also check if we could see the typical aggregates that I showed you before in the patient. So here you have an non-ALS control. So you see these cells here, they don't show any aggregates. When we have a P525L patient without the treatment, you see these things here. This is aggregates, fast aggregates. And after the treatment, the patient rarely show any aggregate, meaning that the treatment was working, but was probably too late. So with that, what I want to show is that fast loss of function is not sufficient to cause ALS. That novel mouse fast knocking lines show motor neuron degeneration and as well as NMJ degeneration, 2 hallmarks of ALS. That the ASO treatment could decrease the fast protein level in mice as well as in humans. The ASO treatment prevents motor neuron degeneration in mice. And we couldn't find any neuronal cytoplasmic inclusions in the patient after the treatment. Thankfully, this treatment or this study is ongoing, so it's already in phase 3. And here there are other patients which were or are receiving the treatment, and it's going to other countries as well. So with that, I would like to thank all my formal lab and all the people involved in this project, and of course, all the families from the patients and the patients themselves. And thank you so much for your attention.
Thank you a lot for a fantastic talk. I think now it's time for the questions. You can either write them in the chat or you can also raise your hand and just ask them to Beatriz. Indeed, I have a couple of questions. So you were showing that there are 2 types of amyotrophic lateral sclerosis, right? The familiar form and the sporadic form. Both of them are really, I guess, are really hard to target, but will any of them will be easier because it's easier to detect the genetics?
Well, the thing is, what we expect is in the familiar cases, because, for example, for this P525L mutation that I showed, it normally, we know the genetic cause, or we have a family history. And then what we hope is that the family familiar people or the family members are tested before to find if they have the mutation and they start the treatment earlier. Because as we have seen, when you have the symptom onset, it goes really fast and it's really difficult that it helps.
And I guess the progression is also really aggressive in both cases, right? Both in the familiar and also in the sporadic form.
There are some cases, for example, as I show Stephen Hawkins, he live very long with ALS. So there are some people that can live longer, depends on the mutation they have. But yeah, it depends. There are more aggressive forms than others.
And then also, do you know something about the clinical trial? Is any hope for other patients who are suffering from ALS? Because I think you have like promising results with the antisense oligonucleotide.
Yeah, there are other antisense oligonucleotide treatments for other mutations in ALS that they are being tested recently. But of course, we don't have, or at least I don't know the outcome from that. So the doctors have to tell this now.
There is also a question here coming from Chi Chung Ling. He's asking, what are your thoughts about the failure of Biogen's ASO trial?
To be honest, I don't know which one is this one. I will have to check because there are so many clinical trials that for ALS.
I guess so. I'm also not familiar with this trial. Yes, if he says yes for SOD.
I will have to check because honestly, I don't know this clinical trial, so I would have to check that. I know they have some other ASO treatments for other neurodegenerative diseases that seem to be very promising. In our case, it's too early to say, so I can't tell. Sorry.
And then so another question regarding the mice models. If I got it right, you do an intracerebroventricular injection of ASO in really young animals, right?
Newborns, yeah.
Newborn. Will it also be possible to do it in the adulthood?
Not in the brain.
Not in the brain, okay.
That's why we can only do one.
Okay.
And so early.
Okay. Very good. Yeah,